Hmm. Okay, can somebody confirm you can hear me?
Yeah, we can hear you.
Great, thank you. I'm not sure if I should wait or continue. There's only two students here. Well, let me start by saying, are there any questions? Let me share my screen here. All right, let me remind you, you need to be starting on your unknown project. This project will take you, well, the average student will take 2 weeks' time to get completed. So you need to get started. All right, I'm not sure what happened. Something weird happened with my Zoom. So let me restart again and say, are there any questions? Thought I shared my screen too. What the heck's going on here? All right. Um, Let me remind you, you need to get started on your unknown project. It will take you at least 2 weeks to get it done for at least the average student. That'll be like requesting 3 or 4 tests. All right, if there's no further questions, let's begin. Can somebody confirm that you can hear me? I'm a little paranoid because Zoom, well, it started all over again. I don't know how to explain it because I didn't even know we lost the connection.
We can hear you.
Okay, great. Thank you. All right, today is October 31st. Does that mean it's Halloween?
Yep.
Yeah, today's Halloween. All right, happy Halloween. Um, we're supposed to be discussing chapter 6, starting chapter 6. We're not there yet. We have to finish chapter 5 first. And there is no lab for today. You should be working on your unknown project and your infectious disease project. We'll be logging in for 15 minutes to answer any questions. But after the first 15 minutes, I'll be logging out. And if you're a little confused on the unknown project, you should log in and ask questions there. All right, any question about what we're doing? Excuse me there. If not, let's go to chapter 5 and finish this chapter. So I talked about in metabolism, you can use carbohydrate degradation or metabolism, catabolism to get your energy needs. And it's frequently, that's the way many organisms get their ATP needs, carbohydrate catabolism. But they can use other means of catabolism, like lipid and protein catabolism as well. And audio there. Zoom just restarted for me. I'm not sure what's going on here, but that's the second time now I've lost Zoom. Can somebody?
Yeah, you did end up cutting out or you were kind of frozen for a little bit, so I did miss a good portion of the lipid protein
I didn't.
slide.
I didn't. Well, this is a review. I didn't say much. I just said that you can digest lipids and protein as well. Let me see. Oh, look at that. PowerPoint shut down even. I'm not sure what's going on. I think it's a problem with my computer. Let me see if I can shut down some things here. I'll need that later, but I don't need it now. I don't need this. Let me shut that down. I'm going to shut that down for time being. Let's see if this works better. Not really. Hmm. Let me shut down a few things here too. Shut down the mail. All right, let's go back to PowerPoint. Blow that up. I tried just switching slides and it wouldn't do that if you didn't notice that. Let me go here. How do I set? All right, there we go. Let me blow that up a little. So carbohydrate, lipid, and protein metabolism or catabolism are all interrelated. I did mention that if you're a couch potato and you're only eating protein, you will burn it. But what you normally do, especially if you're a couch potato, you break down the protein to amino acids. The different amino acids enter um, aerobic respiration at different places. They can join glycolysis fairly late in glycolysis. Some of the amino acids can be broken down into two carbon chunks and then enter as acetyl coenzyme A in the Krebs cycle. Others, amino acids, enter late in the Krebs cycle. The funny thing is I don't have this shown here, but for those of us couch potatoes, we don't directly burn protein. We will actually digest the protein, send it into the aerobic respiration this way, and then go backwards making glucose. And then when we need ATP, we'll burn glucose. But this slide is showing you where proteins can be broken down into amino acids and then will join aerobic respiration in different steps. The same with lipids. Lipids can be broken down into glycerol and fatty acids. Glycerol will enter glycolysis. The fatty acids will be broken down into two carbon chunks, converted to acetyl coenzyme A, and then enter the Krebs cycle there. So all molecules that we eat that are food can enter either glycolysis or the Krebs cycle, regardless of their protein, a carbohydrate, or a lipid. The normal way is, of course, to break down a carbohydrate or just eat glucose directly and then send it through aerobic respiration in the normal way. But as I stated, those of us couch potatoes, the eating a protein or the eating a lipids, we will actually build up glucose and then send glucose down aerobic respiration in the normal way. You can burn lipids directly, and I did mention that a marathon runner, after 45 minutes into their run, they'll start burning lipids in aerobic respiration, meaning you'll break the lipids down into glycerol and you'll enter glycolysis here and then go down the chain that way. Then you'll break the lipids into fatty acids and they'll be broken down into two carbon chunks and then into the Krebs cycle here and generate ATP that way. But they don't do that until 45 minutes into their run. And while they're sitting on the table or a couch watching television, which I meant to say is on the table, they will then break down their lipids and make glucose. And then when they need ATP, send glucose down aerobic respiration the normal way. Any question about any of that? All right, the complete metabolic oxidation of glucose and aerobic respiration is the most efficient. And that's why those couch potatoes like me will change all the food into glucose and then send it in aerobic respiration that way because it's the most efficient. The energy in ATP is then used for various cellular processes, including actual transport of a molecule across the membrane, movement, why my arm is moving, and the synthesis of new cellular components. When we make new molecules, anabolism, we need energy, and that energy usually comes from ATP. So most cellular ATP is actually used for the latter, and that's making cellular components in anabolism, including the biosynthesis of polysaccharides, lipids, and proteins. Any question about any of that? All right. I've already hinted at the integration of metabolism, that anabolic and catabolic pathways are joined by common intermediates. We call a pathway that can function in both directions an amphibolic pathway. They bridge the pathways involved in carbohydrate, lipid, and protein and nucleic acid metabolism, really bridging the catabolic pathways with an anabolic pathway. Animals, including humans, are not very good at amphibolic pathways, but as I mentioned, we can convert, let me blow this slide up. We can convert, I don't have it there. Well, I do it in a way, proteins to amino acids, and then the amino acids we can convert into glucose. And you can see here, like this amino acid, which is converted into oxaloacetic acid, this pathway can go the normal way, but it can go the reverse way in an amphibolic pathway. And if we're going to convert this amino acid into glucose, it has to go the reverse of the Krebs cycle, back to pyruvic acid, back to this ingredient. And this is an amphibolic pathway because there's an arrow going that way, meaning you can go this way to that way, and then up to glucose. And like I said, if you eat a bunch of protein and you're a couch potato, you will not digest the protein directly, usually, to make ATP. You'll convert that protein into glucose. And then when you need ATP, you'll burn glucose anaerobic respiration in the normal way. And the same with lipid. The lipids can be broken down into glycerol and fatty acids, and then they can go backwards to make glucose. So that there is integration of metabolism. Even animal cells can reverse, I guess, aerobic respiration to make glucose. And we're not really good at amphibolic pathways. Animals, including humans, are not good at amphibolic pathways. Green plants and green photosynthetic bacteria are much better at going both ways in a reaction. All right, that's enough for amphibolic pathways. Any question about that? If not, let's talk about the last couple of slides in chapter 5, and that is photosynthesis. Photosynthesis is the conversion of light energy to chemical energy. So you can make ATP by photosynthesis. The chemical energy may be used to convert CO2 to ATP, and oftentimes that ATP is used to make glucose. This we call carbon fixation, where we change atmospheric CO2 to glucose, fixing carbon in the ecosystem. Photosynthesis is performed by obviously green plants, algae, and green bacteria like cyanobacteria, meaning photosynthetic bacteria. It is a mechanism for synthesizing organic compounds, which are required by animals and microbes, which then feed on these photosynthetic organisms. Even if you eat beef, let's say you only eat beef, well, where do you think the cow gets its food from? It gets it from plants. So all living things are dependent on photosynthesis, at least on the land, um, or on chemosynthesis. And we'll talk about that at some later point. Any questions about any of that? All right, photosynthesis only occurs in photosynthetic organisms, and they contain a light-trapping pigment. One of them is known to you. It's green, and it's called chlorophyll. Light releases electrons from chlorophyll, and the light energy is converted to chemical energy via an electron transport chain, meaning we're going to use carrier molecules. ATP and NADPH are generated. NADPH is similar to NADH. And ATP and NADPH are often used up, meaning they're broken down, to synthesize glucose from CO2 and water. And that we call carbon fixation. So photosynthesis always makes ATP and NADPH, but oftentimes the ATP and the NADPH are used up to make glucose. There are actually two types of photosynthetic reactions: cyclic photosynthesis and non-cyclic photosynthesis. All living organisms that I know of can perform both forms of photosynthesis. Non-cyclic photosynthesis is probably the photosynthesis that you guys were taught earlier in Biology 160. A summary equation for oxygenic photosynthesis that you need to know is 6 carbon dioxide, 12 water molecules, plus light generate glucose, C6H12O6, 6 oxygen molecules, and 6 water molecules. Oftentimes you'll see the 6 removed, and then you remove 6 there. But I prefer this equation because you notice all of these 12 oxygen atoms and the 6 oxygen molecules. All of these oxygens come from water. None of these oxygens come from the oxygen and carbon dioxide. So I prefer this equation. Any question about any of that? You do not need to know this, and I'm not going to test you on it, but there's also photosynthesis that does not generate oxygen. It's called anaerobic phototroph or photosynthesis, and it uses sulfur or hydrogen sulfide gas, making sulfur. You don't need to know this one. You do need to know the second form of photosynthesis, and that's cyclic photosynthesis. This is where light energy is captured and then used to make ATP. So in cyclic photosynthesis, all you're doing is adding a phosphate group to ADP to make ATP. As I mentioned earlier, all photosynthetic organisms can make, at least that I know of, I should say it that way, all photosynthetic organisms that I know of can engage either in non-cyclic photosynthesis or in cyclic photosynthesis. Which do they do? It depends what they need. If they need glucose, they'll engage in non-cyclic photosynthesis to make glucose. And you do make ATP in non-cyclic photosynthesis, but the ATP is used up to make glucose. In non-cyclic photosynthesis, all you make is ATP, and the cell will engage in cyclic photosynthesis if it needs ATP. Any question about any of that? All right. In cyclic photosynthesis, called cyclic photophosphorylation, light excites electrons from chlorophyll, right here. Oh, get that out of the way. The electrons then go to an electron transport chain, right here, going to this protein in the chain, going to that protein in the chain, going to that protein in the chain, and then going to that protein in the chain. As the electrons are moving through this chain of proteins in a membrane, they give this protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. The electron then moves back to the molecule it started, meaning the electron will move back to chlorophyll, if this is a green plant having chlorophyll, or an algae having chlorophyll. And that's why it's called cyclic photosynthesis, because the electron travels in a circle, returning to where it started. Now, the hydrogen gradient will have more hydrogen on this side, more positive charge on this side of the membrane than that side of the membrane. These hydrogen ions are going to want to move by the law of diffusion back to this side of the membrane. They can only move across the membrane at this channel protein, which is an ATP synthase protein. And when the hydrogen ions move through this protein, they give the protein the energy it needs to phosphorylate or add phosphate to ADP and make ATP. This should be familiar to you because this is a electron transport chain in a membrane, which is very similar to, let me see if I can go back to it. Which is very similar to the electron transport chain and oxidative phosphorylation or aerobic respiration. Obviously, it's a different chain, it's a smaller chain, but you're sending electrons through this chain of proteins, giving some of the proteins the energy they need to pump hydrogen ion from this side of the gradient to that side of the gradient, and then the hydrogen ions move back to this side, going through this ATP synthase to make ATP. So it's very similar. Any questions about any of that? All right, so where is this happening? In a eukaryote cell, this is happening in a chloroplast. This is the inner membrane of the chloroplast. This is the outer membrane, excuse me, over here will be the outer membrane of the chloroplast, not shown. And so the hydrogen ions are pumped in between the two membranes of the chloroplast. Now, in a prokaryote, which do not have chloroplast, this electron transport chain used in photosynthesis happens across the cell membrane. This will be inside the cell. This will be outside the cell. And so in a photosynthetic bacteria, for example, that's engaging in both cyclic photosynthesis and aerobic respiration, you're pumping hydrogen ions outside the cell into the cell wall. And then these hydrogen ions can move back to create ATP in cyclic photosynthesis, or they can move back to make ATP and aerobic respiration. Any question about any of that? All right. Let's talk a little bit about a summary of ATP production. Let me blow this slide up. The energy source can vary, and that means the electron donor source can vary. The electrons can come, well, they actually come from chlorophyll from sun. The energy source is the sun, and the energy hits an electron in chlorophyll, and then the electrons move. Or the electron can come from glucose if they're engaging in aerobic respiration and they're an animal or something like that. And they can come from elemental sulfur or ammonia or hydrogen gas if the bacteria is a chemosynthetic bacteria. And instead of using glucose for its energy source, it's using some element. It'll strip the electrons from this element. And that will be a chemosynthetic bacteria, which we talked a little bit about. The point is the electron source can vary. Along the way, ATP can be generated. The electrons will be carried by an electron carrier. And we did talk about NAD plus and FAD carrying the electrons through the electron transport chain, where ATP is then made at high efficiency. The final electron acceptor can vary. This slide isn't showing it, but for photosynthesis, the final electron acceptor for cyclic photosynthesis will be the same molecule the electron came from, meaning chlorophyll, for a green plant. And that's not shown here. I'm not sure why it's not, because they have photosynthesis there, and so they should have it down here, and they don't. So that could be a final electron acceptor, the chlorophyll of cyclic photosynthesis. You can also have oxygen being the final electron acceptor, and that's what aerobic respiration has, is that its final electron acceptor. For anaerobic respiration, we have a final electron acceptor being a molecule other than oxygen, like nitrite or sulfate. And then for fermentation, the final electron acceptor is a molecule in glycolysis and the fermentation steps, meaning it'll be an organic compound where the electron flows to. Any question about any of that? All right, that's it for chapter 6. Chapter 5, let's move to chapter 6. I have to open that again. I closed it down because my computer was doing funny things and it shut down Zoom twice. Fortunately, Zoom started up again, but still, it's annoying. Let me shut down chapter 5. All right, in the first slide of chapter 6, discussing microbial growth, I list the major goals and a rough outline. Always know the terms. And remember, if you want to know all of the objectives, go to the study notes of chapter 6. 2, understand the various requirements of organisms for growth. 3, understand the 4 phases of growth. And 4, compare the different methods of measuring microbial growth. Any question about any of this? All right, let's move on. Microbial growth. When we're talking about microbes, remember that we're talking about growth. We're talking about the increase in the number of cells. We're not talking about the increase in the cell size. I used to teach middle school students, and when we were talking about them growing, we were not talking about them reproducing. Any questions about any of that? Well, this is being really slow. I'm trying to show you microbes growing.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
All right, any questions about any of that? There are requirements that the cells need for microbial growth. The cells have physical requirements and chemical requirements. Physical requirements include things like the correct temperature, the correct pH, the correct osmotic pressure, meaning the correct salinity. Chemical requirements include they have to have a carbon source, they have to have a nitrogen source, they have to have a sulfur source, they have to have a phosphorus source. They need trace elements. They may need oxygen, but if they don't need oxygen, then they may require oxygen to be absent. And then they have organic growth factors, which may be required. Can anyone mention an organic growth factor for humans? Something that we require in our diet because we can't make it. And obviously, it's a chemical source for something. Anyone know an organic growth factor for humans?
I don't know, like proteins, like nitrogen.
Well, that is true. We do need protein. That's usually for a energy source, but you could require it for nitrogen and carbon, in which case it would be a carbon source. So that is true. That's not really an organic growth factor, and I'm not sure why. Um, Probably because the cell can make proteins, assuming it has the starting subunits from amino acids. The cell can make protein, but protein is something that a cell needs, but it's not an organic growth factor. Nor an organic growth factor is something you must have in your diet, otherwise you'll become sick and eventually die. And that would be something like vitamin C. Humans cannot make vitamin C, so we must get it in our diet. And that would be a small molecule we have to have in our diet. And for humans, that would be an organic growth factor. Any question about any of that? Now, for bacteria, if they need vitamin C, they can make it themselves, at least most of them. And I don't even know if they need it, but they might. For the physical requirements of temperature, each species has its minimum temperature, its maximum temperature, and its optimum temperature for growing. The optimum temperature will be when the species can grow at its fastest. The minimum temperature will be the minimum temperature that it can grow at. And if you go lower, the cell will not grow. It may not die, but it will not grow. And of course, the maximum is the maximum temperature the cell can grow at. Microorganisms are classified into groups on the basis of their preferred temperature range. So here we have psychrophiles, and they grow best at 10 degrees, well, 4 degrees to 10 degrees, we'll say it that way. These are organisms that grow in your refrigerator. They cannot grow in you because they will fall apart if they get to that temperature, but at least some of their molecules will fall apart and their enzymes won't function at your body temperature, but they can grow in the refrigerator. And it's because of these organisms that any food you put in the refrigerator, unless it has a natural preservative or maybe an artificial preservative, you should throw it out after about a week. Because after a week, these psychrophiles can grow to large enough numbers to spoil the food. So that's why all food you put in the refrigerator should only be left in the refrigerator for about a week. and then it should be thrown out, unless it has its own natural preservatives. Like ketchup has both acids in it and high salt and high fructose or sugar, sorry, in which case it can stay in your refrigerator for years. I know my mother had a bottle of ketchup that was over 2 years old, and I told my mother, you and I aren't using this. I was living with her at the time because I was sort of taking care of her when she became elderly and unable to take care of herself. But I told her, we're not using this ketchup. It's over 2 years old. Why don't you give this to my brother? He uses ketchup and goes through it like, I don't know, anything. And of course, we gave it to him, and a month later, he used it up. Probably less than a month. The point is that it had preservatives in it, and it could stay in the refrigerator for longer than 2 years and was still good. Then there are psychotrophs. These are organisms that grow best around room temperature. And then there's mesophiles. These are organisms that grow best around 37 degrees Celsius. That is human internal body temperature, 98.6 degrees. All human pathogens are mesophiles. Any question about any of that? Thermophiles are organisms that grow best around 60 degrees Celsius. And if you ever see a chicken at Safeway or someplace, and they've got it cooked and they have it under the heat lamps, and it's rotisserie going around and around, and you look at it and it looks kind of old and looks kind of dry, the thermophiles can actually grow at that temperature. So if it's starting to look old, probably best not to buy the chicken. Although I think they would probably not grow to high enough levels within a day to, what's the word, contaminate the chicken or make the chicken go bad. I'm not sure how long they would take though. It would speed up because it's a week in the refrigerator, and that's 10 degrees Celsius or less. And so it would be less than a week because it's 60 degrees Celsius. The organisms will grow faster, but I don't know how long the chicken can be in the rotisserie under the heat lamp and still be good. I just don't know. And then there's a hypothermophile. These are organisms that were not known when I took microbiology. These organisms grow best above 90 degrees, so around 95 degrees Celsius. Remember, 100 degrees Celsius is the temperature that boiling water grows at. Any question about any of that? Now, these, the average for each of these is not rigidly defined. Like the average mesophile grows best at 37 degrees. But if you talk about Micrococcus luteus, which grows best on our human skin, and I guess Staphylococcus epidermidis the same, it will grow best around 30 degrees Celsius. We'd still call it a mesophile because it's close to 37. And then especially the temperature range, meaning what the minimum temperature is and the maximum temperature, that can vary too for each species. So it's around 10 degrees and around 50 degrees, but it's not exactly going to be 10 degrees and 50 degrees, meaning there is a range for each of these classifications. And it just depends on the species, what the range will be. Any question about any of that? Talking a little bit more about a hypothermophile. The Hispanics, like a Mexican, if they have a volcanic vent in their neighborhood, the Mexican women will put corn in a gunny sack and then throw it in the volcanic vent, which will be close to 100 degrees. Allow it to sit there for, I don't know, 10 minutes or so. Pull the gunny sack out, and then take the corn home and eat it. And the hypothermophiles have never made them sick. So as far as we know, there's no hypothermophile which causes human illness. That doesn't mean that there aren't. I'm just saying that as far as we know, there aren't any. Any question about any of that? Oop, not too far. Food can spoil if the food is kept at the wrong temperature for too long. So the danger zone to keep food at will be something like between 60 degrees and 130 degrees Fahrenheit. That would be about 15 degrees Celsius to 51 or 52 degrees Celsius, where the bacteria can grow rapidly at this temperature. And it can grow to high enough levels that some of them may produce toxins, and then the toxin will make the food unedible. And so you need to keep the food out of the danger zone, out of the temperature danger zone for, well, less than 4 hours, for example. Anything longer than 4 hours, you probably shouldn't eat it, meaning if you keep your food, what is it, between 15 degrees and 50 degrees, 52 degrees Celsius, for over 4 hours, you probably shouldn't eat it. You can have bacteria growing at a slower rate at a lower temperature, at least down to, I don't know, what is that, 5 degrees Celsius, up to maybe 61 degrees Celsius, where the bacteria can grow very slowly, and which, given enough time, can make the food unedible. The principle for preventing food spoilage is to refrigerate the food in small enough batches so that the food cools quick enough that it goes through the danger zone quick enough and the bacteria do not grow to high enough levels and then to make a toxin. Because at this point, there's not going to be enough bacteria to notice the food is gone. So it won't be a problem unless the bacteria are making a toxin. But if you refrigerate it in too large of a batch, the food will stay in the danger zone for too long. What is that? 1, 2, 3, 4, about 5 hours. And that will be long enough for Bacillus cereus, for example, to multiply in rice and then make a food toxin. Any question about any of that? So you want to take the rice, for example, and put it in the refrigerator where it cools at below the danger zone in about an hour or less. Another physical requirement is pH. Most bacteria grow well between pH 6 and pH 8, meaning they grow best around pH equal to 7. This is a problem for lab cultures because when we're growing microbes in the media, they often make acidic microbial end products, which can then inhibit growth. To minimize this problem in the lab, we add a pH buffer to the culture media, and then the pH buffer will buffer the pH, preventing the acids from changing the pH. And then you can grow the microbes for a longer period of time. Now, molds and yeast grow best between pH 5 and 6, so they are a little different than most bacterial cultures. And then there are some microbes which are acetophiles. They grow best in acid environments less than pH 2. A volcanic environment can reach as low as pH 0 to pH 3. And the stomach, when it's producing acids, can be pH 2 or less. So we do have acetophiles growing in our stomach. Helicobacter pylori is one. If you don't know, Helicobacter pylori can cause ulcers. Any question about any of that? All right. Microbes do have some physical requirements like osmotic pressure. Microbes require water, and they have to grow under the correct osmotic pressure necessary for the water to stay in a cell. If the cell is put in let me show this up. If the cell is put in an isotonic solution, there's no net movement of water into the cell, and so the cell is happy. But if the cell is put in a hypertonic solution, water will leave the cell, and then the cell membrane will shrink, pull away from the cell wall, causing plasmolysis, which can inhibit growth and may even kill the bacteria. Like E. coli, for example, is not very good at surviving in a hypertonic environment. And after a certain amount of time, I don't know, about a day, maybe only a few hours, I don't know, but not very long, E. coli will undergo plasmolysis and then die. Obviously, it depends on the cell how long it can survive in a hypertonic solution. Staphylococcus aureus can survive longer than E. coli. But even Staphylococcus aureus, like when it falls into a bottle of molasses, it will eventually undergo plasmolysis and then die. The cells which can survive the highest in a hypertonic solution is, of course, a cell that has an endospore. And it could survive for, well, centuries, millennia in a hypertonic solution. But it will not grow. It can survive, but it will not grow. A hypertonic solution can happen when there's too much salt in the environment of the cell or too much sugar. In molasses, it would be too much sugar. High osmotic pressure can be used to preserve food, such as the salting of fish or the salting of meat, drying fruits and vegetables, causing high osmotic pressure, and then the food won't spoil. Molasses being another example. Any question about any of that? Now, you can have a halophile. This is an organism that likes to be around salt. We have two types of halophiles. An obligate halophile requires a high salt concentration to grow, like an organism growing in the Dead Sea. That's at 30% sodium chloride. The Dead Sea is between Palestine and Jordan. It has obligate halophiles in it because 30% sodium chloride. The only thing that can live in that would be an obligate halophile or a facultative halophile. A facultative halophile is an organism that does not require a high salt concentration to grow, but can tolerate salt higher than 2%. And some of them can grow in 15% sodium chloride. Staphylococcus aureus is a facultative halophile. It does not require high salt concentration to grow, like it can grow on a nutrient agar plate. But if you grow it at a plate with 15% sodium chloride, Staphylococcus aureus can grow. And Staphylococcus aureus can survive up to 20% sodium chloride, and that would be the highest salt concentration your skin would get. Depending on when you last had a shower or a bath, your skin could climb up to 20% sodium chloride. And the opposite range is in a hypotonic solution. This is low osmotic pressure, where there's lots of distilled water around, or water around. Water can enter the cell by osmosis. This may cause the cell membrane to balloon and expand until it pops. Unless the cell has a cell wall, a cell in a hypotonic solution will eventually pop and die. And that usually happens just within minutes. Like you put red blood cells in water, distilled water, within a few minutes, all the red blood cells will pop and die. If the cell has a cell wall, that will prevent the cell membrane from expanding any further, and that will prevent water from moving into the cell because there's no room for the water to move into the cell. And that can keep the cell from lysing. And that's why most bacteria have a cell wall. All right, another chemical requirement of cells is they have to have a carbon source. Carbon is typically half of the dry weight of a cell. So remove all the water from a cell, half of the weight remaining is from carbon. Carbon is used in structural organic molecules. It's used as an energy source. You can find carbon in the membrane, in many of the molecules in the cytoplasm. Heterotrophs are organisms that use an organic carbon source. So we are a heterotroph. We mainly use as our organic carbon source glucose. An autotroph is an organism which can feed itself, so that means self-feeder, and it can use carbon from CO2 dissolved in the air or the water. So they get their carbon from CO2, an autotroph. Any question about any of that? Some other chemical requirements include nitrogen. Nitrogen is 14% of the dry weight of a cell. Nitrogen is in amino acids, in proteins, DNA, RNA, ATP. Nitrogen is often obtained from the decomposition of protein. And many bacteria can get their nitrogen from protein. But many bacteria can use ammonia or ammonium or nitrate as their nitrogen source. A few bacteria can even use nitrogen gas from the atmosphere. And these organisms we call nitrogen fixing bacteria because they can do nitrogen fixation, pulling the nitrogen out of the air and then making it available in the ecosystem. Like when the bacteria die, that nitrogen pulled out of the air will then be available for other organisms in the ecosystem. Any question about any of that? Sulfur can be obtained from amino acids, from thymine, that's a B vitamin, biotin, also a B vitamin, and from, sulfur is required for DNA and RNA synthesis. Sulfur can often be obtained from the decomposition of proteins, and that's usually where humans get their sulfur source from. Many bacteria can get their sulfur from decomposing proteins, but many can also get sulfur from sulfate ions. We cannot, or from hydrogen sulfide gas. Hydrogen sulfide gas is a toxin to us, so it would be toxic to us. But some bacteria can actually use, many bacteria can use sulfate, and some can use hydrogen sulfide gas as their sulfur source. E. coli can use sulfate, and E. coli can also obtain their sulfur from decomposing proteins. Phosphorus is required for making DNA or RNA or ATP. Phosphorus is also required for the phospholipids, which make up the cell membrane or any membrane. Phosphorus is often obtained as phosphate, so most bacteria can get their phosphorus source from phosphate. And humans can actually get their phosphorus source from phosphate as well. Any question about any of that? All right. Carbon is something that all organisms require. It's typically half of the dry weight of the cell. Carbon is used in structural organic molecules. Obviously, carbon is the main molecule holding the organic molecule together. Carbon is also used as an energy source. Heterotrophs use organic carbon sources like glucose. Autotrophs use carbon dioxide as their carbon source. I already talked about that. What happened there? Oh, here we go. Trace elements are something else that are required by all living life forms. A trace element is usually an inorganic element required by microbes in trace amounts. They're often required as enzyme cofactors. For example, iron, copper, and zinc are enzyme cofactors. And we must get these trace elements in either something we eat or something we drink. And that's true for microbes as well. You can usually get trace elements in your environment. One exception, if you're only eating food grown in the Western Washington or Western Oregon, or you only drink liquids made in Western Oregon or Western Washington, you won't get enough iodine because all of our rain has leached iodine out of the soil. So we have to have an iodine source, and you can get that from iodized salt. But all the others, trace elements, iron, copper, zinc, we get from our diet or something we drink. I know copper you can get from dried fruit like raisins. I'm not sure where you get zinc from. Iron you can get from red meat. If you cook in an iron skillet, you can get it from the iron skillet. You can also get iron from certain vegetables, like I think broccoli, maybe green leafy vegetables. I don't remember. I should know, but I don't remember. If there's a what do you call that? A vegetarian here, you can pipe in where you get your iron source from, other than an iron capsule or a multivitamin. All right, an organic growth factor is also a chemical requirement. If the organism requires an organic growth factor, you must get it in your diet. Organic growth factors are small organic compounds obtained from the environment that the organism cannot synthesize itself. So certain vitamins or organic growth factors. For humans, all of the vitamins except for what is it, vitamin A and D, we have to get from our diet. A and D we often get from our diet, but especially in the summertime, you can make them with exposure to sunlight, although you have to have the precursors. But sunlight can make, I think it's vitamin D from a precursor. But like in the wintertime, you probably need the vitamin in your diet. There are certain essential amino acids and certain essential fatty acids that we cannot make. And so we call them an essential amino acid or an essential fatty acid. We need to get these essential amino acids and essential fatty acids in our diet, like the amino acid lysine is an essential amino acid for humans. We cannot make that amino acid, so we need to get it in our diet. There are some amino acids, I think it's phenylalanine, that we can make. And so it would be a non-essential amino acid. And then other examples of an organic growth factor are purines or pyrimidines. Certain microbes require a purine or a pyrimidine in order to grow. And if I think about it, I'll actually name one that is, but I'm not getting its name off the tip of my tongue now. It's not Streptococcus pyogenes. What the heck was it? It's a human pathogen. Requires purines and pyrimidines to grow. And it gets it from us if it's a human pathogen. All right, oxygen is also a requirement for cells. If they require oxygen, it's absolutely required. If they do not tolerate oxygen, then it's required that oxygen is absent. So molecular oxygen is, in a sense, a toxic substance, including to even aerobes, organisms that require the presence of oxygen. There are certain oxygen intermediates which are toxic even to an aerobe. And there are 4 toxic forms of oxygen. The first being singlet oxygen. This is oxygen gas, where an electron is boosted to a high energy state. This singlet oxygen is highly reactive. Singlet oxygens are actually present in phagocytic white blood cells, and they'll put it in the lysosome, and then they will engulf something in a phagosome, and then fuse the lysosome with the phagosome so that that singlet oxygen can come onto what you've phagocytized, let's say a parasitic bacteria, and then kill the parasitic bacteria with the singlet oxygen. Any question about any of that? So cells have to remove singlet oxygen when they make it, and singlet oxygen is always made at very low concentrations in aerobic respiration. That's true of almost all of these toxic forms of oxygen. The second toxic form of oxygen is superoxide free radical. This is oxygen gas with an extra electron attached to it. It's a free radical because it only has one extra electron, and it's a -1 from that extra electron. And the molecule does not like being a free radical with one extra electron. It either wants to give up that extra electron or it wants to gain another electron. And so a free radical will bump into almost any other molecule and then steal an electron from that other molecule. And that's the danger of free radicals. And that is they bump into things and then damage the thing. And then when it steals an electron from whatever it bumps into, let's say it bumps into, I don't know, a ATP molecule, you steal the electron from ATP, that ATP then becomes a free radical because it's missing one electron. That symbol there refers to an unpaired electron, which is found only in free radicals. Free radicals are highly unstable. They are formed normally during aerobic respiration. And then these free radicals steal an electron from whatever they bump into. And then that other molecule will bump into another molecule, stealing an electron from it. And that goes on into the cell. And that if it bumps into DNA, it can damage the DNA, including create a mutation. And that's the danger of free radicals. Free radicals can be neutralized by the enzyme SOD. So here we have a oxygen free radical, actually 2 of them. SOD can combine to oxygen free radicals with two hydrogens to make hydrogen peroxide gas and oxygen. And that's why we have SOD in our cells, to remove superoxide free radicals. Now, hydrogen peroxide itself is a toxic substance to the cell. So hydrogen peroxide is a toxin, and we have to have some way of removing this hydrogen peroxide. Hydrogen peroxide is a peroxide anion right there. And cells have two ways of removing peroxide anions, including hydrogen peroxide. They can use the enzyme catalase, changing two hydrogen peroxide into two water molecules and oxygen. Hydrogen peroxide can also be broken down by the enzyme peroxidase, where it takes one hydrogen peroxide, two hydrogen ions to make two water molecules. All cells that live in oxygen have to either have catalase or peroxidase or both enzymes, or else they will be killed by a peroxide anion, such as hydrogen peroxide. Any question about that? And I guess that's true of actually all aerobes. They have some means of removing all of these toxic oxygen molecules. We have SOD in our cells, and we have catalase in our cells. There's also the hydroxyl radical. This is a hydroxide group with one unpaired electron. Why it's got that symbol there. It differs from the hydroxide ion, which has a negative charge to it and two electrons, so that it doesn't need to steal any more electrons. And the hydroxyl radical will not want to be one extra electron. It'll want to either give up that electron or it will want to gain another electron. So when it bumps into another molecule, the radical will steal that electron, and then that'll change from a hydroxyl radical to a hydroxide ion. Oops, didn't mean to change that. Hydroxyl radicals are always made by aerobic respiration in trace amounts. They can also be made by ionizing radiation or when hydrogen peroxide reacts with certain metal ions. How cells control and remove hydroxyl radical is they have antioxidants that combine with and remove the hydroxyl radical. Two antioxidants that we have to combine with hydroxyl radical and then remove it are vitamin C and vitamin E. They can combine with the hydroxyl radical and then remove it. Unfortunately, that does remove the vitamin C as well, but it's better to remove the vitamin C and then remove the hydroxyl radical, because the hydroxyl radical can bump into DNA and then mutate it. Any question about the four toxic forms of oxygen? All right, microbes that use oxygen and aerobe must have oxygen around to make ATP. And then we have microbes which cannot tolerate ATP, not tolerate oxygen, and they do not use oxygen. So they are an anaerobe. Microbes are classified on the basis of the effect of oxygen has on their growth. What you do is you put the cells in a thioglycolate tube, The thioglycolate tube is a media that removes oxygen from the media. And when you first take the tube out of the autoclave, it will have no oxygen in the media, but oxygen will come into the media from the air, so that the top of the tube will have full oxygen from the air, and the bottom of the tube will have no oxygen. And as you go down the tube, you'll have a gradient of decreasing oxygen. At the bottom of the tube, you have no oxygen. At the top of the tube, you'll have full oxygen. And that is a thioglycolate tube. Any questions about that? You then put the microbe that you want to test in the tube. If the microbe only grows at the top of the tube, that would be an obligate aerobe, an oxygen that requires, did I say an oxygen? I meant a microbe that requires oxygen to grow. So it'll grow best at the top of the tube. And as you go down the tube, you'll have less and less growth because there will be less and less oxygen. At the bottom of the tube, you'll have no growth because there is no oxygen. And that's an obligate aerobe. The next easiest is an obligate anaerobe. At the top of the tube, where there's complete oxygen, you'll have no growth. At the bottom of the tube, where there's no oxygen, you'll have growth, and the most growth at the bottom of the tube. As you go up the tube, you'll have less growth. There should be, oops, let me go back. There should be one dot right there, meaning you have a little bit of growth in this region here because there's less oxygen. And so the microbes can grow a little bit, but if ever they bump into an oxygen, they'll then be killed. So they can grow a little bit in this region because there's less oxygen. And then they can grow a little more here because there's even less oxygen. At the bottom of the tube, they have their full growth rate because they have no oxygen. And that's an obligate anaerobe. The next easiest is a facultative anaerobe. These are organisms that can grow in oxygen, but they can grow without oxygen. They grow best at the top of the tube. And as you go down the tube, you'll have less growth. Why do they grow best at the top of the tube? Because they can perform aerobic respiration at the top of the tube, because there's lots of oxygen. And they grow best or make the most ATP when they perform aerobic respiration. But down at the bottom of the tube, there's no oxygen, and they're falling back on some other means of making ATP. It could be anaerobic respiration, it could be fermentation, doesn't matter. The point is at the bottom of the tube, there's less growth. And as you go up the tube, there's more and more growth because there's more and more oxygen. And at the top of the tube, you have the most growth. And that's facultative anaerobe. There's also an aerotolerant anaerobe, and these grow at the same rate at the top of the tube, the middle of the tube, and the bottom of the tube. These organisms are anaerobes, so they do not use oxygen, but they have all of the enzymes and molecules they need for growing when oxygen is present. So oxygen does not inhibit their growth. And there's only a few organisms which are aerotolerant anaerobes, but they can grow everywhere the same in the thioglycolate tube. And the last category is the microaerophiles. These are aerobes, so they do require oxygen to grow, but they don't grow best at full oxygen, the top of the tube. They grow best someplace in the middle of the tube where the oxygen level is reduced. Elobacter pylori is a microaerophile. It grows in the stomach where the oxygen level is between 2% and 10%. And the point is they'll grow best in the middle of the thioglykylate tube. They don't grow best at the top of the tube. They don't grow best at the bottom of the tube. In fact, they don't grow at all in the bottom of the tube. Any question about any of this? How are we doing on time-wise? Oh, let's just introduce the topic of media. We've talked about general purpose media. Remember, medium is singular, media is plural. You can have a liquid or a solid media. A liquid we call a broth. A solid media has the broth with something added to it to make it solid. Oftentimes in microbiology, we add agar to the broth to make it a solid media. But you could add something else like gelatin or starch to make it a solid. The media you get in the lab will be sterile, and that means it has no living microbes in it, including endospores, by the way. An inoculum is what you add to the media to get the microbes to grow. So when you add microbes into the tube or onto the plate, that would be our inoculum. The culture is the microbes growing in or on the culture media. So you got a tube, and I say it's a stock culture. That's what you're supposed to use for your future experiments. Like in theory, I gave you a stock culture of your unknown bacteria, and I told you what the number was, and that's your stock culture. Although you should streak it out just for pretend sake, meaning you're pretending you do it, and then you write up your report. You say, I was given this culture and I streaked it out for colony isolation 2 times to make sure that the culture was pure. We have a very wide variety of media available in the microbiology lab. I could show you a book about this size. It's actually a little bigger and about this thick. On each page of this book, we have one media. And that tells you how many different media we have in microbiology that we could use to grow the microbes. Any question about any of that? All right, if there's no questions, I think I'll stop here. And there is no lab for today, but if you have questions, I'll be in the lab for the first 15 minutes. Any questions? All right. I will see you next time.